This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 151, recorded September 29th, 2011. Hi everyone, I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today, after a long absence, from Western Massachusetts, Alan Dove.
Good to be here.
Haven't heard you in a while, have we?
Yeah, it was one darn thing after another, wasn't it?
Well, we had two remote recordings, and
yeah,
then,
you hit the road for a while, and then
missed you last week.
Yeah, daughter got sick.
Too bad. It was 153-year anniversary.
Yeah, exactly. I listened to the episode. Actually, I've gotten about two-thirds of the way through the episode, but I said, oh, man, I should have been on there. But
happy birthday.
Yeah, the microbes had other plans.
That's okay. Everything's okay?
Oh, yeah. Yeah, she's fine. And antibiotics, miracle drugs.
So far, they're miracles.
Yes. Let's so let's all not try to screw that up, okay?
Go listen to TWiV.
Yeah.
16 was the last one, I think, on antibiotic resistance. Man, it's pretty scary.
Yeah.
Also joining us from North Central Florida, Rich Condit.
Hi, fellas.
How you doing there, Rich?
I'm doing really good. I've been looking forward to this, all day long. I'm figuring I get to kill an hour and a half at the end of the day just talking with you guys.
It's good. It's a lot of fun.
That's good.
Yeah.
Unfortunately, Dixon can't join us, even though he's right outside my office. He's waiting for 20 minutes from now when a Korean TV crew comes to interview him. I think they're building a vertical farm there. So.
Yes, as I said,
Yep.
I think there's something in this episode about that.
So
but,
he is
we'll see.
waiting for them, and I thought he'll just sit here with his phone and wait for the call, so I thought it'd be better if he didn't start with us.
Yeah.
And today we're going to finish all the email we didn't do last time. We have a lot. I would love
we'll
to.
Be lucky to finish it,
we're not
actually.
going to finish it.
We'll make some progress.
We'll make some progress, so this will be a true all-email episode. The other thing I wanted to mention is that I spent the last few days at the Scientific Advisory Board meeting of the Chronic Fatigue Immune Dysfunction Syndrome Association of America. So I'm spending time working on this disease, and so I'm concerned about it. But I met there many patients with the disease, and to each one they recognized that XMRV is over. They were patients, and they were parents of children with the disease. And again, all of them understand that XMRV and CFS is finished. So, okay.
How was that meeting? That
was
must
a
have been
great
very
meeting.
interesting.
The best part for me was meeting people with CFS.
Yeah, I would think so.
A variety of mainly people in their 30s and 40s who had started careers and then were disabled and can't work any longer or can only work part-time. I met a woman who is in fact a fan of TWiV who started working as a lawyer and was disabled by CFS and now is on disability, and many other individuals who can't work or do the things that they want to do. The interesting thing is they can remember the day that it began with an infection of some kind. Mm-hmm. And so maybe any kind of infection triggers an immune response that is abnormal, and figuring out what that is what we have to do. I met a number of physicians who treat CFS patients. Dr. Nancy Klimas in University of Miami was one of them. She was there. And also Dr. Cindy Bateman from the University of Utah. Both very dedicated physicians who care about these patients and have got clinics up and running and also doing clinical research to find out what's going on. So the CFIDS Association is trying to sort out how to support the best research, how to raise money, and then how to support the best research to find the cause and eventually a cure.
Cool.
So I'm happy to participate in this, and I don't get paid for this. It's my time I'm giving to this organization. And I was introduced to this organization through blogging and podcasting. They realized that we are dedicated to this, so I was asked as a virologist to join. And I might add, I also met Dr. Robert Silverman, who has been on a number of these XMRV papers, and in fact, who authored the retraction that we discussed last week.
Yeah.
All right. So
a real gentleman, Bob.
He's a good guy. Actually, he says, hi, Rich.
Thank you.
And he is a very nice
High
fellow.
back. His
And
brother works upstairs here.
that's what he told me, yes. Yeah. And, you cannot tell what a person is like through emails.
No.
Not at all.
No, I've always had the greatest respect for Bob.
Bob's a good guy. So he's also a member of the board, and we're going to meet periodically and try and figure out how to crack this. It's not going to be easy. I think it's quite a complicated disease, but I am very impressed by its effect on these individuals. There are also parents there whose children have the disease, and they're very active in trying to figure out what's going on. So I think we actually should have an episode on CFS where we get a couple of the patients to talk about it, and we talk with the CFID CEO and find out how they're dealing with it.
That'd be great.
I think we should wait for the last bit of the XMRV thing to die down first.
Yeah, probably after the...
maybe sometime next year after the Lipkin paper comes out.
I have to wonder, with all of these samples now that are being collected in a very controlled, blinded fashion, et cetera, for the Lipkin study, I have to wonder if a deep sequencing effort on those things might not turn out.
Well, he's going to be doing that for another foundation, I believe.
So there is another foundation called the Chronic Fatigue Initiative, and you can find that on the web, CFI, and they've given a large amount of money to Columbia, Harvard, Stanford, and Miami. And here at Columbia, they are recruiting 200 patients and 200 controls. Ian Lipkin will be deep sequencing and looking for pathogens. They're going to be establishing a biobank of samples for use by other people. So it's not going to be any one foundation that's going to do this. So it's good that a second one has come into this.
Well, that's good. And as we all know now from all of this experience, having a number of different laboratories involved and taking a number of different approaches are important for getting at the real truth.
Yep.
Yes.
I understand, by the way, that another paper is imminent. It is not the Lipkin paper. It's something else. So it'll be out in the next couple of weeks.
All right.
We'll be telling you about it. All right, let's do some email. First one is from Stephen, who writes, in response to Dixon's question, was it last episode?
It was an email that we did last time because Dixon had misspoken. He called horses ruminants.
That's right.
Okay? And then as we were discussing that last time, Dixon asked whether zebras and various other animals, and he may have mentioned wildebeest, were also not ruminants or whatever.
So
there's a name for the horse version. Monogastrics.
Yeah, monogastrics, one stomach. That's right. So Dixon said, what's a wildebeest? And I said, well, let Stephen answer. So Stephen did answer. He said, wildebeest are ruminants as they belong to the family Bovidae, like the domestic cow, etc., whereas zebra belong to the family Equidae and are monogastrics. Again, a great show, Stephen.
It's always good to ruminate on the news.
I didn't know about this.
Hey, was that news to you, Rich?
It was news to me, yeah.
Wow. All right. Alan,
good.
Why don't you take the next one?
Sure. Christos writes, I have two comments about your latest episode. One, you read a letter by a neurologist about virus reactivation during immune modulator therapies, and he asked you for examples. You mentioned herpes reactivation during natalizumab therapy. His comment is correct, and I guess we'll see many more of these cases in the coming years when more biologic immunomodulators are used. Currently, the most common example of a virus reactivation is hepatitis B during all kinds of anti-TNF treatment, including several antibodies. There are several reports out there that in some countries there is an active monitoring of hep B levels during treatment.
I didn't know that. Did you?
I didn't know that either, no.
That was news to me, too.
And I'm really impressed with how natalizumab rolls off Alan's tongue. I really struggled with it.
I'm not sure that's the correct pronunciation, but
it sounded good. You said it with confidence.
Yeah,
right? That was good.
I'm frequently covering things that end in zumab or izumab,
So the Hep B thing, no, I did not know about that. That's very interesting. And all this, makes you have to wonder about mechanism, and I don't know.
Yeah,
very interesting.
But
obviously, immunomodulators are going to do things to, especially. Hep B is a chronic infection, right?
Right.
So,
I tell you, they're going to be trying immunomodulators for treating CFS. That was one of the things I learned. So it could be one of these monoclonals, in which case then you have to be careful for
have to be careful
for Hep
right.
B and PML.
Absolutely.
And also know in advance what you're trying to treat.
Yeah.
Because you can't just start medicating just based on immunological markers.
Yeah.
There has to be some scientific basis for why that's supposed to work. Okay, and then continuing with Christos's letter. In a lighter note, you mentioned Bachman's comments on the HPV vaccine. And he links to a video in the Colbert Report making fun of the whole issue. And it's a typical Colbert take on it. It's pretty good.
Christos is actually a former student in this department who is now working at a local biotech company.
Cool.
Thanks for that, Christos. Okay, Rich, next one.
Judy writes, finally, I can help you guys. PBS has a great evolution series and an associated website, and she gives the link. The videos are good, may need additional info to update. So this is in response to a letter we read last time where somebody had written in wanting some sort of video or something on evolution that his wife could use in her teaching, and was frustrated by his inability to find anything. Dixon recommended going to some museums, and she writes in with this PBS evolution series. She says, and then there is the Understanding Evolution website from UC Berkeley School of Paleontology, and she gives a link to that. So glad I get to give back. Judy, who describes herself as a high school and community college person. P.S. Yes, please to the Virology 101 podcasts, and keep up the bad jokes and puns.
All right.
We've kind of let Virology 101 slide. We need to make some time.
We're on transcription. It's right.
By the way, I discovered that a postdoc in my laboratory has been listening to your online lectures, Vincent. Mm-hmm. Right? You got a complete set of lectures there that are available anytime you want them. And she said, as much experience in virology as she has, she never had a real virology course. Like me, I've never had a real biochemistry course, and I taught in a biochemistry department for 12 years. And I've never even had a microbiology course, and I'm in a microbiology department now. Who knows?
Yeah, they're on iTunes, and they're also being uploaded to YouTube. Columbia has a YouTube channel, and they're slowly putting them up there. So I'll put the link to that when they're finished. All right, the next one is from David, who writes, Viri Guys. In episode 139, in response to a letter from Eric, Vincent at about the hour mark made reference to a paper detailing evidence of viruses contained in genomes extending back about 150 million years. Vincent stated he would put a link in the show notes, but I didn't find it there. Would you please put in that link? I would be interested in reading this paper. I think it's cool. When I forgot to put it in, I said, nah, nobody will notice. And it's great that someone picked it up. So I have a link to a blog post where there are all the references, 5 or 6 different references. And I will put that in these notes as well as the original.
So people actually do follow the links.
I do.
Mm-hmm.
In episode 141, in part of the discussion about Alan's pick of the week, there are a few things to add or correct. The Permian was from 300 to 251 million years ago, not 100 million years ago, as Alan stated.
Yep, I screwed that up.
Rich asked, what about what kinds of animals were around, and Alan mentioned Dimetrodons, the sail-backed animal that looks like a dinosaur. A Dimetrodon fun fact, we as a representative of mammals are more closely related to Dimetrodons than dinosaurs. Dimetrodons are part of the class Synapsids, referred to as proto-mammals, from which mammals evolved. There were also gorgons, which had big canines, sort of like saber-toothed cats. Some of the first marine reptiles existed then. All of the landmass was part of a single supercontinent named Pangaea. The Permian ended with the largest mass extinction event, the mother of all mass extinctions, when an estimated 90 to 95% of all marine species and 70% terrestrial species went extinct. By comparison, the last big mass extinction, the one that got the dinosaurs, only rated a puny 75% species extinction. We actually talked a little bit about the KT extinction on TWiV 16 at ICAC.
Right.
These mass extinctions are just hard to think about, It's amazing.
Yeah. Yeah, the end Permian extinction is particularly mind-boggling.
What was
And
supposed to have caused that? Do you know?
well, we don't exactly know. The current theory, and there's some evidence to back it up, is that there was a major volcanic eruption, I think in Siberia, what's now Siberia or thereabouts, and it spewed vast quantities of CO2 and other gases, and I guess reduced oxygen, so a lot of things died. But it's what's so to me mind-boggling about it is 95% of marine species and 70% of terrestrial species just gone.
Yeah.
And it's this event that in the fossil record it happens in the blink of an eye. It's just there's this incredible, and the Permian was full, as the blog I was talking about points out, was full of just these amazing animals and this incredible biodiversity that is not really even been seen since. There was this whole thriving ecosystem on the planet, and then just bang, it's gone.
I always find this stuff, in its own way, reassuring, because DNA will replicate. You
Yes.
can wipe out you can wipe out almost everything, and it comes back in some form.
But at the same time, you look at that and you say, our planet is capable of doing that.
Yeah.
That's cool. Well, Arturo Casadeval has a theory about the KT extinction and how fungi wiped out some species and so forth. So go back to TWiV 16. Check that out.
Okay.
David also has a pick of the week, which I'm going to save for another time because we have a pick for this week. So we'll move that to another time.
Yeah, that looks like a good one.
All right, before we go on further, I want to acknowledge the support of TWiV by Wiley Blackwell, the leading scientific publisher of books, scholarly journals, major reference works, and databases. This month, they're offering 25% off of all microbiology and virology books. To take advantage of that offer, go to wiley.com/go/microbeworld. That is wiley.com/go/microbeworld, and you will find their microbiology and virology catalog, which is pretty neat. They have a lot of good titles, and get that 25% off. If you're on Facebook, you can go to their facebook.com/microbiology news, all one word, fan page. And we thank Wiley Blackwell for their support of TWiV. I believe Alan is next.
Yes, so Jim writes, "Hi, Vince et al. I heard a TED talk by Angela Belcher on using using nature to construct batteries, fuel cells, etc. Viruses are used to move genetic material to bacteria to make materials like abalone and diatoms make. I vaguely recall a pick of the week for viruses being used in batteries. If the Angela Belcher talk is different from this, it may be of interest. I'm a devoted fan of TWiV, TWiP, and TWiM. It's a privilege to be able to hear you and your colleagues talk. 73, Jim. Oh, right, Jim's a ham radio operator.
Oh, what does that mean?
That means best wishes.
I thought he was telling us how old he
regards.
was.
No.
Cool. Good to have you on, Alan. Well, we have talked quite a bit about making various things from viruses by Angela Belcher and others. The TWiV 98, 28, and 8, viral batteries. And remember, there was one where they were weaving it into army clothing.
Right.
Yeah, right.
Using these filamentous viruses. And then TWiM 14, we talked about microbial transistors using bacteria, not viruses. So go check those out, Jim. We have talked about them. It's very cool. She's always in the news about doing this stuff. Rich.
Dear virologists, may I try again on Heisenberg? One of the first things a student in physics learns is that we probably know less than 5% of everything there is to know about the universe. And further, they learn that we probably never will know all there is to know, or even a significant portion of what there is to know. Indeed, there is even one point of view that the human brain, at its current level of development, may not be capable of understanding certain things, but that's another topic. This leads to a certain level of modesty on the part of physicists about their power to understand any topic. Did I detect a certain sense on your part that virologists are gradually learning all there is to know about viruses, and eventually there will be no uncertainties? Yes, Heisenberg's use of uncertainty is different than the uncertainty of undiscovered phenomena, but in the larger philosophical context, the point is that no matter how much we study something, it is very, very unlikely that we will ever understand it in every detail. And I wonder if the more you study and understand viruses, you uncover more questions than you do answers. What do you think? I forgot to mention that my training is in relativistic quantum mechanics, but have become an amateur virologist. By the way, the virology I work on is of dengue. Your TWiV 147 on dengue and Wolbachia was superb. What do you think are the chances that dengue will become a more virulent virus as it tries to evade Wolbachia in mosquitoes? Are there any precedents? And this is from Rich, who is from the Dengue Vaccine Initiative in Seoul. Have we heard from him before?
Yes, yeah, we have several times. Yep.
Right. Okay. Yeah, he's written us letters about that initiative.
I'm not sure I'd describe him as an amateur virologist if he's coordinating policy for the dengue vaccine initiative.
Right. Well, one of his comments about the vaccine initiative that I found a little puzzling as well, if I remember correctly, is that, in fact, although they're coordinating this initiative, it's more of an administrative task than a real basic science task. So they have a lot of people who don't have a lot of virology in their backgrounds who are working on this. And that's one of the reasons that he appreciates TWiV and recommends it to his colleagues.
That's right.
Yeah.
The more I learn, the dumber I get. There's no question about that.
Yeah, I don't think we said that we are getting to the point where there will be no uncertainties, right?
No. I think that was intended sarcastically, wasn't it?
Probably.
And actually, one of the things that frustrates me is that, I was just thinking about this today, is that the field sort of, there are these fads that happen, or, there seems to be a herd, a front line of science that kind of moves along, and they haven't necessarily finished what they did, okay? So, case in point, you used to go to ASV meetings or virology meetings, and it was all about genome replication, transcription, gene expression, all the basic nuts and bolts. And then pathogenesis became the thing. And I'm not saying that that's bad, it's good, okay? But everything is pathogenesis and immunology now, and you don't hear anything about genome replication, transcription, and stuff. And that stuff's not done, all right? We still don't know anything about a lot of the basics, but everybody's kind of moved on. So not only are we never going to understand everything, but there's a lot that's unfinished, a lot that has been studied that's unfinished.
Yeah, and the amount of unfinished stuff is expanding.
Yeah.
When you look at the petabytes of data coming out of some of these deep sequencing efforts, nobody can explore all of that right away. And so the lab that did the original deep sequencing picks something, and they go after that. And the rest of the rest of the data are just sitting there, and they're hoping somebody else will pick it up and do something with it. So yeah, the more we study, the less we know.
Yeah.
Yeah, I certainly feel the same way. There's no question about it.
And
about that last one, yeah. Go ahead.
So I don't think it's necessarily going to become more virulent as it tries to evade Wolbachia. I think that, generally variation has a tendency to reduce virulence in these critters.
It depends on what it needs to change in order to evade Wolbachia.
Mm-hmm.
Yeah.
And, as was discussed on the episode, it's going to happen. The virus is going to do something. You apply a pressure and something's going to change, but it's an open question what.
Yeah, typically there are fitness costs when such things happen. So
right.
If we had to predict, and even though it's kind of dangerous, I would say it's not going to become more virulent.
Right. I can't think of any precedents. He asked for precedents. I can't.
I don't. Yeah. You know any, Alan, where you've put a natural control in and the virus gets around it?
I don't even know of a precedent where we've really implemented a natural control.
But there are probably some examples in plants or fungi that we don't know about. So if anyone knows.
Yeah, I mean, there are some natural control implementations that manage to locally eradicate something.
Yeah.
But didn't necessarily apply a selective pressure because it successfully eradicated the thing in that area.
Right. Okay, thank you, Richard. The next one is from Derek. Hello, TWiV. I love your podcasts. I'm a PhD student in Victoria, BC, and drove to San Francisco and back this summer and spent the whole drive catching up on past TWiV episodes. I think I'm saturated. That's pretty cool to think of someone driving up and down the coast there listening to TWiV.
Yeah.
I would get saturated listening to that much TWiV.
But you're not saturated doing it, right?
No,
this is fun.
In podcast 146, you had a pick of the week from the do-it-yourself biospace, which happens to be what I was in San Francisco for. There's a new community lab called BioCurious that some of your listeners may be interested in. It provides a space to explore wet lab biology in a more casual setting than pursuing an academic degree. It's at biocurious.org. Also, Ari Gentry and I have just launched a podcast of our own that may interest your listeners. Garage Bio is a podcast devoted to exploring what happens when you put the hottest new biology discoveries in the hands of amateurs and small-scale startups. GarageBio.org.
Cool.
You're an inspiration. Keep up the great podcasts. Derek, anybody listen to Garage Bio?
I didn't listen to it. I looked at the website. They're just on episode number 3 or something like that. They have interesting backgrounds, so my guess is that it's an interesting discussion.
All right, check it out, folks. I've never heard of it, so thank you, Alan.
Okay, so Lily writes, to all who make TWiV happen. Thanks so much for all the great information on your netcast. I'm a non-scientist, newly introduced to your program, and I'm tuning in every week now. It's an absolute treat to listen to scientists talk in an absolutely fascinating manner about such a vast array of topics having to do with virology. TWiV 149, where you chat with Trina Sideris, was a real eye-opener for me in particular, and I've taken the opportunity to read several of Trina's informative articles that you've linked to on the program. Cheers, Lily.
Well, that was nice.
Yeah. Yeah, we like those.
Yeah, that was a good episode with Trina.
Yes.
That was fun. Is it me?
I think so, yeah.
Dear TWiVsters, I am a UK listener and, having recently discovered your excellent podcast, have been doing my best to catch up. I am not quite up to date yet, so apologies if you've already discussed this, but I would be really interested to hear what you guys think about the potential for phage therapy to support or even eventually replace antibiotics. I found this topic fascinating and looked to pursue it both as an undergraduate in biology and during my MPH. Almost as fascinating is that none of my professors ever seem excited by its potential. It would be great to hear your views on this topic. Keep up the good work, Peter.
Do you notice how everyone is addressing us in a new way?
Yeah.
This is TWiVsters, to all who make TWiV happen. Cool.
Viri guys. So you had a TWIM on this?
Yeah,
Vincent?
we did. In fact, Michael Schmidt, one of the TWIM co-hosts, has worked on this.
Oh, okay. Well, we've touched on it a couple of times in TWiV, but we've never really done it total justice.
So, Peter, go over to TWIM numbers, microbeworld.org/TWIM number 6. We talked about it, and those guys are experts. I don't remember who else was on the show.
And what does Michael think?
So Michael thinks that you need a cocktail, which is tailored to each patient. So he thinks you have to get the bacteria, identify phages that are going to kill those bacteria, and then you give that to the patient. Wow. So he says there's no other way to do it because everyone's got a unique set of strains in them, right? And the one that's causing disease in you may not be the same that's causing disease in someone else in terms of phage susceptibility. That is a problem for regulatory because you can't regulate something you make on a patient patient-by-patient basis. So that was a lot of the discussion on that show.
Yeah, that's partly, that would certainly be true depending on the amount of engineering you do. But if you look at something like Provenge, this ferociously expensive prostate cancer therapy from Dendreon, it is a custom therapy.
Oh, yeah?
And it's done on a patient-by-patient basis. It's one of the reasons it's expensive. The other reason it's expensive is, of course, they have market exclusivity. But it's they actually get cells from the patient, bring them back to their lab, and work with them, and then take them back to the patient. And there's this whole logistics that goes with it. And part of getting that approved was really kind of pioneering through the regulatory process for a custom therapy that's built that way. But the point is, it's not easy to do.
So there are companies both here in the U.S. and in other countries that are working on this.
Yeah.
You have to listen to that episode. But as on TWiV 16 in ICAC, they only mentioned it briefly, but they basically said it's not working yet. So it's not there, but we'll see.
Still an idea that people haven't totally given up on.
No, yeah.
Right.
All right, the next one is from Arnold, who addressed this to Rich. Dear Rich, in one of the last TWiV shows, you said that HIV is the only RNA virus which integrates in the host genome. This is not true. You might have missed the papers about Bornavirus or Ebola virus, which showed that these viruses can, or at least did, partly integrate in the genome of its host. So, did you miss these papers, or do you have a different opinion on that? So when I received this email, I immediately replied that Rich and us, we said this because for HIV, integration is an obligate part of the life cycle. For the other examples, the Ebola and Borna, which we are well aware of, as evidenced by our previous email above, it's not obligate. So then he replied, I did not want to say that it was not an accident, and it is also not obligate for these viruses, I know. But it has happened and maintained and still might happen, and it also seems not to have a disadvantage neither to the host nor to the virus. One can even turn this and think about if this might have had a beneficial effect for one or both, even though it is not an obligate feature. As always, when people start to dig in things, it turns out that there is not only black and white, but also gray. Maybe this is the only thing I wanted to say. Okay. Yes, I mean,
I'm all about gray.
Yeah.
We discussed about whether these were beneficial or not, these integrated Ebolas and Bornas, because they're still there. It's an interesting question for sure. Right. But Rich's statement, and correct me if I'm wrong, was just saying that's the only one where you need integration. Retroviruses.
Well,
right?
Certainly that would have been my intent. All right.
The RNA viruses.
And I make this distinction in class all the time. There's lots of different viruses that wind up getting their nucleic acids tangled up with the genome, but it's an accident. I mean, the classic is, in my mind, is human papillomavirus, where integration into the genome is not a normal part of the virus's life cycle, but when it does, and does so in the in an unfortunate configuration, the result is cancer, right?
Yep.
So, yeah, you stick a nucleic acid in the nucleus, in particular DNA, and the cells are kind of stupid. They say, "Oh, that's DNA. I know where that goes." And boom, it integrates.
All right, Alan.
So Jason, who goes by the nickname of Xenophage, writes, in a previous TWiV, 126 to be precise, you talk about the HPV vaccine. Again, if I understand correctly, you mentioned that if someone is infected with HPV, they don't generate antibodies, but that injection of the HPV vaccine does cause generation of the antibodies. I believe the question of why was asked, but there was no known answer. Is it possible that with a full-on infection, the virus is able to somehow evade the immune system, effectively preventing the creation of antibodies, but the vaccine is an attenuated form of HPV, allowing the immune system full access to the virus? Just some random speculation from a non-virologist.
Well, it's more than random speculation. That is a great question, and that's one that I've puzzled over for a while, and so I'm glad he brought it up.
Yeah, and I guess, Vincent, you forwarded this to Michelle Osborne, right?
That's right.
So here's her reply. She says, this is a good issue requiring clarification. Only a fraction of those with detectable HPV DNA will show an antibody titer, suggesting that a small dose might be able to infect but not generate a measurable immune response. Yes, we know that during infection the virus evades the host immune response quite well. Exposure to the virus does not normally result in inflammation, which is important for an immune response. The skin is an immunologically privileged site, and the virus is not lytic and escapes via shedding of dead skin cells. Therefore, antibody titer is not a good measure of whether someone has been infected previously. Viral DNA or sometimes viral transcript detection is the measure of current infection. Conversely, the HPV vaccine is a pretty high dose of a subunit-type virus-like particle preparation with an adjuvant, alum, that is injected intramuscularly. The virus-like particle cannot replicate because of the high dose, route of inoculation, and the adjuvant, which causes inflammation, and the fact that the major capsid protein, L1, is present in repetitive capsameric structures, 360 copies of it arranged in 72 pentamers, the vaccine elicits a strong protective antibody response. The antibody response shows efficacy and protection well over 5 years of follow-up. Hope this helps to clear up the difference in exposure and immune response.
Nice.
Yeah,
that's
nice,
excellent.
Nice response. Yeah.
So HPV sneaks in normally, and the vaccine is it's not a live virus. It's a virus-like particle. It's a shell essentially, plus an adjuvant, and you put a huge dose of it in. It can't sneak in and it can't replicate. It's just this massive dose that the immune system sets off all kinds of alarm bells for and generates antibodies against.
And the key here is inflammation. So
right.
What we're talking about here is the infiltration of immune cells into the infected area, production of cytokines, which are all needed for a good immune response.
So it's interesting. Actually, we probably ought to expand this conversation with Michelle. So the one ordinarily mounts an immune response to papillomavirus. Most of the infections are self-limited. Okay, they disappear. By the way, this virus causes warts, and it also causes skin lesions on mucosal surfaces like the cervix, and a rare outcome of this is cervical cancer. But in most cases, it causes warts, and they're a self-limited disease, and they just clear up and go away. So there is an immune response. Okay, but it doesn't show up as high titers of serum antibody. Okay, that's not very reproducible at all. As a matter of fact, that's why there aren't, not only that, but you can't grow the virus in culture, right? So that's why we don't have serotypes, right? In order to have serotypes, you have to have a virus and serum to compare them. Well, we can't grow virus. Well, now we can make recombinant capsid proteins, but when the serology was trying to be done, we didn't have virus, we didn't have serum, so you couldn't make serotypes. So it's all based on nucleic acid. At any rate, you don't make a good antibody response, and yet you do overcome the infection. My understanding is that a lot of it is cell mediated. Okay, though I may not be entirely accurate in that sense. But it's interesting that even though it's as if the immune response that is triggered by the virus-like particles is different than the normal immune response you have to the virus. But it's effective. It takes care of it.
Right.
Yeah. It's a good question, Xenophage.
It's a great question.
Yes,
good
it's an
job.
excellent question.
You're listening. I think he's been listening to us for a while. Good job. Rich.
Greetings, TWiV crew.
Hold on here. Is this the wrong email? Maybe it is. Yeah. This is for TWiV. Sorry. Go on to the next one.
You are a busy guy. Levi writes, "Hi, Vince. It was great to see you at ASV. I just wanted to note that I'm currently watching Ian Lipkin on Charlie Rose. He's there speaking about the film Contagion, which is about a deadly viral pandemic. Like you, I'm often disappointed with the portrayal of science in film and television. I know exactly what you were talking about when you mentioned NCIS on your TWiV about zinc finger nucleases. Because of this, I don't usually watch such shows or films because I know they'll get it wrong, and I won't be able to suspend disbelief and enjoy the work. However, since reading a couple of good reviews and now seeing a respected scientist involved with the project, I'm tempted to see the film. I'd be interested to know if you or the rest of the TWiV team have seen it, If so, what are your impressions? I, for one, would love to see a film that accurately depicts the work of those involved with infectious disease and public health. As virologists, we know many true stories that are more thrilling than any Hollywood script. Hantavirus at Four Corners comes to mind. But maybe as virologists we are biased. Maybe people who are so fascinated by reality find fiction a bit disappointing or unnecessary. Thanks for all the great TWiVs so far. Keep up the good work, Levi.
It's great. Fiction is a bit disappointing.
Yes.
Yeah.
I haven't seen it yet. You have, Rich. Have you?
I
Alan?
have seen it.
I have not.
Because when we all see it, we'll do a show on it. How's
Right.
that?
That
Right.
sounds good.
My lips are sealed until then.
All right. But he's
Except to
right.
say, I do know that a lot of other people in my lab and my environment have seen it and really enjoyed it.
He is absolutely right. Virologists are biased. So when you see a film about your field, you're very critical.
Yeah.
Because you know what the reality would likely be.
Mhm.
And that's why it's hard for us to see these things. But we'll reserve judgment, Levi. Levi, by the way, is in your department, right?
That's correct. He's right down the hall.
Cool. All right.
I think it's your turn, Vincent.
Yes. Oh, Alex writes, hello, fellow TWiVzors.
That's a new one.
Now, what is a TWiVzor?
That's you and us, I guess.
Yeah.
Cool. I apologize in advance for the wide spectrum of this email. Been meaning to write for a while now. Let's see here. I'd hope that Dixon would be for this part. Well, I'll read it anyway. I enjoyed TWiP 29, Neglected Tropical Diseases, with Peter Hotez. It was very insightful to hear about the neglected tropical diseases and how mortality rate is not always the clearest way to measure the impact of a particular disease. I was wondering if you might be able to inquire him on why he selected the MD-PhD program in his education. I've only recently become aware of this route offered by many medical schools and wondered if he might talk a little on the advantages and disadvantages. While 7 years seems like a rather large commitment of time, it appears as though it blends both treatment and novel research well. Additionally, I'm also curious as how it's best to approach a faculty member when looking for a mentor for a research project. Reading their current fields of research online is a good start, but I'm not exactly sure where to go from there. Most of the medical schools that I've been investigating require you to have a clear idea of what you intend to research and with who at the time you apply. Actually, we can answer this ourselves.
I think so.
Who wants to talk about why you would get an MD PhD?
Well, I'm not necessarily the best one to talk to because I'm not sure I would get an MD PhD. There are people around who can be physician scientists, okay, but it's a rare bird. I mean, it's two jobs, both of which are more than one job, and so being a successful physician scientist is, boy, it's tough to do both well. I think a good thing to do. Well, you can do an MD PhD. You can also go to medical school. If you really, you go to medical school if you really want to treat patients.
Yeah.
Right? If you really want to do research and you don't really care all that much about patients, you go do a PhD, right? And neither one prevents you from participating in the other. After all, I'm a PhD who's been working in medical schools for 30 years, and I have plenty of opportunities, as do my PhD colleagues, to get involved in transitional research. Conversely, you can be I know plenty of MDs that are quite capable researchers. You don't necessarily need both degrees. A really good route, I think, is to get an MD and then go do a fellowship at a place like NIH or something like that and get training in research. You don't necessarily need the degree. Not that a good MD PhD program is a bad thing to do. Right?
Yeah, I would actually suggest picking which one you want to do. I've talked to a lot of translational researchers and certainly in an academic setting, there's this idea of MD PhDs who they get a certain amount of protected time to do their research, and it's 80% research, 20% medicine, or 50-50, or they've got some things spelled out in a contract. But every one of them who have asked about that off the record has said, no, they absolutely don't protect that time. There's just you get asked to do MD things while you're trying to work in the lab, and your lab work, somebody you've got to deal with some issue with it while you're trying to treat patients, and they're trying to, as you pointed out, it's two more than full-time jobs, and you're trying to do both of them.
I think we have had MD PhDs on TWiV before, Rich, because I know you always comment how tough it must be to juggle clinical and research responsibilities.
God, it's hard to imagine. There's the rare few around who manage to do both jobs completely, but boy.
So let me give you an example here. We call these individuals physician scientists,
right? And
for the most part, they see patients a month out of the year, and the rest of the time they do research in their labs, and that's what they're required to do. It depends on the specialty, of course. It can be more than that, but that's very typical. Now, if someone who is doing clinical research, where they have to see patients all the time, might do it more often. But that's an example of when you might want to have both, if you want to see patients and treat them and then take their material and bring it into your lab and study it at a basic level.
Yeah, and if your clinical responsibility really is restricted to only a month a year, okay, and outside that, there's really not much clinical responsibility. But we really have to get an MD PhD on the show to talk about this. But my guess is that in that circumstance, you're still a member of the clinical faculty.
Right.
Okay.
Sure.
So even though you're only seeing patients for a month a year, you probably have responsibility to your clinical faculty that goes outside that.
Now, having said all that, there is one thing that an MD PhD degree definitely does for people, and that is it drastically increases your chances of qualifying for a highly competitive residency program.
Mm-hmm.
And I know people who have, now you don't want to tell the medical school that on the way in, but but that it does have that effect. There are people who do it, and then they end up going into surgery, and they are surgeons. They're not doing research in the lab, but they qualified for a good surgery residency partly on the basis of having had the dual degree.
Yeah. Now, just to echo what you said, Rich, I know many faculty here who have an MD only. They see patients once a year, and the rest of the year they do research because they did a fellowship of some sort and learned how to do it.
Right.
So you don't have to do both.
You don't need the degree to learn how to do the research.
Yeah.
You do need to go to medical school to learn how to be a doctor.
That's
okay?
Right.
Yeah.
But you don't necessarily have to go to graduate school to learn how to do research. You can learn that just by being thrown into a laboratory. And I would also say, even though I am not an MD, I've got some experience in this. I'm on the medical selection committee and that kind of stuff, and hung around medical schools. I think that if you want to be an MD, it's got to be because you want to treat people.
Yes.
Yep.
You want to see patients. You want to be with the patients. All right.
And that's not just because medical schools will expect that of you. It's because if you don't genuinely have the drive to treat patients, you are going to be miserable in medicine.
Yep. Now, his last question there, most of the medical schools I've been investigating require you to have a clear idea of what you intend to research and with who at the time you apply. I don't believe we do that here.
No, that's not the case here.
So, but I think if you go in with some good ideas and you can discuss it at your interview, that would be fine. Because someone's going to ask you, what kind of research are you interested in? And you should have looked at what the faculty does, read about the research, and pick a few that you like. In fact, sometimes you can pick specific research faculty to interview with. And I often do that. They have an interview with one clinician and one research faculty. And you can read up and say, I'm interested in this stuff. Even if you're not going to work on it, at least it shows that you had some interest and you read about it. And I can tell you that not everyone does, and it's not good when you come to an interview and you don't know what anyone does.
Yeah, and bear in mind that it's what you say in the interview is not legally binding for what you eventually choose to do for research.
Right. Mm-hmm. Absolutely.
So what you're interested in now is just, figure out a clear articulation of why you want to do an MD PhD. and articulate that in the interview. And then if you decide later that you want to work on something entirely different for your research years, then that's...
And I would say, in answer to his question, how is it best to approach a faculty member when looking for a mentor for a research project? Just directly contact them.
Yeah.
Email, telephone, walk into their office, whatever is possible.
Yeah. Send them an email, say, hey, I'm interested in your work. Can I come talk to you? That works.
Right. And if they say yes, that's good. If they say no, that's good too.
Yeah, move
or
on.
If they don't respond, move on.
Move on.
Right, exactly.
All right, he continues. Regarding TWiV 147, I was slightly saddened that the quintessential theoretical endosymbiote was not mentioned. You guessed it, the mighty mitochondria. I know several studies have suggested this organelle was once an independent bacteria that may have fused with a eukaryotic cell at some point in its lineage, perhaps one of the oldest examples of mutualism, where the host eukaryotic cell gains ATP energy while the bacteria gains protection from the outside environment. How would one explain the presence of unique RNA within the mitochondria otherwise? Anybody know about mitochondrial origins more than I do?
Not any more than what he's written right here.
Yeah, that's pretty much what I know about it. It's either the oldest example of mutualism or the longest running case of indigestion.
Yeah, we weren't being comprehensive, but you're absolutely right. It is a good one. Also, is there a such thing as an exosymbiote? Benign bacteria of the gut falling into this genus of Bacteroides, Clostridium, and Fusobacterium seem like they could be labeled as this. The host is protected from more virulent species while the bacteria gets a free source of nutrition. Sure, you could call those exo. They're not within us, within our cells. Sure.
Right, and topologically they're contiguous with the outside of us.
And Dixon reminded me of, for example, bacteria in the gut of ants that produce antifungals. And this is a really interesting story, which we will do either on...
It sounds like a TWiM story.
Yeah, probably TWiM.
Yeah.
So these are leafcutter ants. You know, they use fungi to digest the leaves, and the bacteria in their gut produce antifungals that keep away the wrong fungi, basically. It's so cool.
Amazing.
Right. It's another example of an exosymbiote. And then finally he writes about contagion.
And we got to save that because he's got
he's got great stuff here.
A whole bunch of stuff about the movie.
Yeah, he writes, I'm glad to see the movie.
You shouldn't even read this. You need to see the movie first. Don't read this.
I'm not going to read. I'm just going to read his first paragraph, okay? I'm glad to see that the movie Contagion didn't fall under TWiV's radar, as it was selected in a weekly pick on 148. And then he lists a bunch of spoilers, basically. And we're going to save this for our Contagion episode.
Right.
Thank you for all the diligent work on TWiV. Episode 150 will soon be here. Quite a milestone, to say the least. Thank you, Alex.
Yeah.
Alan, you are next.
Okay, so Charles writes, Hello, TWiVers. In a recent episode, there was a passing mention of the fact that for a cell, a defeated virus is also a source of nucleotides. I was wondering if there are any organisms that deliberately encourage virus penetration, confident that they can neutralize the viruses and then use them as a food supply. Or is this too dangerous a strategy to have evolved? It seems to me, from my dubious position as a software engineer whose knowledge of biology is largely due to your podcasts and Wikipedia searches, that a possible candidate might be a bacterium living inside another organism, luring in viruses that infect its host. As the viruses would have evolved to infect the host rather than the bacteria, inviting them inside might not be immediately suicidal. Thank you all very much for the work you put into making TWiV. I never miss an episode. May you long continue. Kindest regards. That's an interesting idea. It seems like it would be a very risky strategy. And the thing that actually comes to my mind is the general lifestyle of macrophages, which do kind of make a living this way. They go around eating stuff that's not supposed to be in the bloodstream. Now, of course, they're our own cells.
Or amoeba, which are free-living macrophages.
Yes.
You know, their whole job is just to eat garbage.
Yeah.
I'm not coming up with any
No, real examples.
examples of this. No.
No.
Yeah, but deliberately encouraging virus penetration is not a particularly good survival strategy, I would think. Because you'd be better off just eating the virus. It's not as if viruses are not swimming around. You don't have to lure them.
Yeah.
So if you swim up to one and it's made of protein and nucleic acids and you're an amoeba, then that's lunch.
Yeah.
Not a very big lunch.
No.
It's a good thought. But,
Yeah, it's a neat idea.
I don't think it happens, but who knows? I believe Rich is next.
Jeffrey writes, Dear Dr. Racaniello, recently you edited a PLOS Pathogens paper, The Fecal Viral Flora of Wild Rodents. And I had a question that doesn't need an immediate answer, but perhaps could be addressed in a future TWiV. The authors isolated these viruses through filtration, treating the filtrate with DNase and RNase to nix any unprotected nucleic acids. I found it interesting that 40% of their reads were bacterial and 2% were eukaryotic. Are these what you would expect from schmutz sticking to the filter? Do you think it's possible that these represent phage that are caught mid-transduction in the case of the bacterial sequences, and maybe eukaryotic viruses doing the same? Just dreaming like a second-year graduate student. That is the question. Now the plea, are there any plans to do a program on the various papers that have come out on viruses in the feces of different animals? Ever since graduate school working on adenovirus at Emory University, I have wanted to look into daycare poopy diapers to address questions of adenovirus excretion and sequence evolution. Now I am a full-time teacher at a community college near Atlanta, but I find myself working with others in my department, trying to get a shoestring program together to have our non-majors students collecting coyote poop so that we can extract DNA for sequencing, to have students look at real-world local applications of what they're learning in class. So far, all I have is several coyote scats stored in the freezer and am looking at what exactly we should be amplifying for sequencing. It's a tougher problem than I initially thought it would be. Forgive me if you've already done a program like this. I will get to it. But you guys really serve as a weekly, sometimes bi-weekly lab meeting journal club for me and help keep me not only current but excited to bring hot off-the-press science to my students who would otherwise not likely think about these things.
Are coyotes local to Atlanta?
I wouldn't be surprised if there were coyotes around there. We got coyotes around here.
Okay.
The pepper model virus in human feces story would be a great way to get Senor Despommier into the studio, as it brings up issues that he specializes in, relationships between different species and the poop agriculture pathogen / parasite triad. I would love to hear his opinion of whether he would consider humans to be a vector for this virus. I consider the communication of science and how it works to my non-science major students to be one of the most important things I do, and you really inspire me. We may try to do a simpler version of what you do, but with the focus on the amazing diversity of plants and insects that we have around our rural campus. Thank you for a very entertaining and patently useful program. Jeff. So apparently you wrote to Eric Delwart about this.
Mm-hmm.
Was Eric a co-author on this paper?
He was, yeah.
Okay. So Eric replied, great question, which I'll rephrase as why we don't get 100% viral sequences since we purify for capsid-protected nucleic acids. So they filter this stuff and treat it with nucleic acids, so they're trying to get stuff that is strictly viral. No extracellular, no free nucleic acid, nothing that is retained in a filter, okay? Just small capsid particles. But they get all this bacterial and eukaryotic sequence. Lots of the bacterial and eukaryotic hits are to ribosomal sequences, which we reckon are partly protected from nuclease digestion due to their being in a tight complex with proteins. Ribosomes are also about the diameter of a small virus and can easily go through our 400 nanometer filters. Our definition of viral is also quite stringent at an E-score of less than a lot of zeros and a 1. So we might actually have more divergent viruses than we report. Lots of the sequences had hits to phages, so the proportion of viral sequences, both prokaryotic and eukaryotic, is actually 13%. I reckon a lot of that background is due to bacterial, protozoal, host DNA, and RNA being in some form of small schmutzy complex with proteins, lipids, glycoproteins, and surviving the nuclease treatment. We did notice that these rodents had a very low ratio of eukaryotic virus hits to total sequences relative to what we saw with other mammals like bats, sea lions, and pigs. I think this reflects a generally lower enteric viral load. This may be due to a relatively more isolated lifestyle for rodents and possibly the older age of the feces donors. A lot of the sea lions and pigs were less than a year old. So, schmutz is the basic end. Ribosomes and other things that, despite all of their best efforts to get rid of the extraneous nucleic acids, creep through this.
I think a show on viruses and feces would be good. It's a little early. It's not that much yet. But once we have more, I'm sure Alan could come up with a cool name for that.
A good title. I was exactly thinking, yeah.
Okay. Thank you, Jeff.
Feces happens.
Feces happens.
When the virus hits the fan.
Next one is from Peter, who writes, as a longtime listener of TWiV, TWIP, and TWIM, this is to Dixon. You and Vincent almost seem like old friends to me, thus it feels somehow special to find your name and your vertical farm project mentioned in the large German periodical Der Spiegel.
This is the thing I was talking about.
That's right. Yeah, he's got a link to an article in German, but you can, of course, translate it readily. Let me take this opportunity to thank you for the work you're doing. The way you're shedding light on microscopic life and not-so-life is the best educational entertainment and the most entertaining education I know. My amazement about the interwovenness of all life has grown beyond my wildest beliefs, and I'm sure I've only just scratched the surface. Wow, I could use that quote for something.
That's cool.
This is great. Nice. Thank you, Peter.
And I think this article is an article in Der Spiegel about vertical farming, and I think that one of the farms they're highlighting here is in Korea.
Oh, really?
I think. I'm having a little... At any rate, there it is.
Cool.
The best educational entertainment and the most entertaining education I know. That's a great blurb.
I like it. Can use that in our grant applications.
Yes.
All right. Alan, I believe.
I think it's my turn, yes. So Jason writes, hello, TWiV. I'm a physicist pretending lately to be a biologist, BS in math and physics, now attempting an MS in biology. I was looking for new podcasts recently to listen to for a road trip and came across TWiV, starting at around number 132. I must say it was a very educational road trip indeed, and have been enjoying trying to catch up from TWiV number 1. It's been a few months now, but I remember a conversation about HPV, and some of the talkers were from UF down here in sunny Florida. Just wanted to see if they caught the HPV article UNF student newspaper published around that time. Caught some very interesting publicity off their choice of cover picture for the article. I thought it was an interesting grab to educate the students at UNF that HPV might not only be a female-type problem and the dangers in oral sex, and found it funny that I heard a comment on TWiV just after that about throat cancer risks with HPV. Take care, and hopefully sometime this year I'll catch up before taking my first virology class. My project will have something to do with oral inoculation of arbovirus in mosquitoes and finding the little pests very interesting, both of them. Oral inoculation of arbovirus in mosquitoes, that's got to be a very small tongue depressor.
All right.
So we talked about this problem of oral sex-derived cancers transmitted by HPV in TWiV at ASM. So we had Dr. Katzenellenbogen talk about that. Yeah, that's an interesting cover there.
Yep.
Yeah,
that would get college students' attention.
Yeah, that would get your attention. I don't think it's probably suitable for the TWiV picture.
No, I don't think so.
No, I don't think we can illustrate the episode with that.
No, I'll get more hate mail. Oh.
Right.
Where is University of North Florida, Rich?
It's in Jacksonville.
Okay. All right, Rich.
Steve writes, "Hi, Alan and Rich."
Wow. Oh, that's your episode. That's why.
Yeah. Superb job co-hosting TWiV 145. You didn't miss a beat. Just a small bookkeeping note. The hyperlink to the listener pick of the week is currently incorrectly to the smallpox and its eradication PDF article and not to the Science Photo Library webpage. Thanks for fixing this. I see you did fix it.
Yep.
Keep up the good works. I so enjoy your show and look forward to the Sunday download, which I listen to on Sunday evening. Warm regards, Steve. Steve is an MD. That's good.
Cool.
Actually, I'd like to hear from some, if there are physician scientists out there who are listening, I'd like to hear their responses to that email we had about physician scientists a little while ago.
Yeah.
I'd like to get more input into that.
Yeah.
Yep.
Yeah, clearly none of us are experts on the issue.
Right. Absolutely.
All right. The next one is from Dave. Now, I think I read part of Dave's email before. I don't remember.
You've got this. You've got it twice here.
So Dave had written about the Draco antiviral, and he wanted our reaction to it, and we've done that. And I forget the TWiV number, but by now you know that, Dave. So that's that. And then the reason I wanted to keep this in here is that he mentions he's an associate professor of biology in Pennsylvania. I wanted to share that this spring I had my undergraduate virology students use TWiV to learn about potential pieces of literature that they could then download and read as the basis for their in-class oral presentations. Overall, the students liked hearing a little context about the paper from TWiV before going through it themselves in detail to prep for their talks about the experiment's data. An added bonus of the exercise was that a number of them are now regular TWiV listeners. So I just wanted to read that as another example of how teachers are using TWiV in their classes, because we always get questions about how you can integrate it a little better.
Cool.
All right. Now, Alan, the next one is yours. And
all right.
I would truncate this substantially.
Yeah, this goes on a ways.
But just give us the, you could read the first couple bits and then give us the gist of it.
Let's see. So Robin, who's an MD, writes, Vincent and Dixon, listen, I love you guys and don't want to get anyone in trouble, but someone should address our government's preoccupation this last decade with the development of offensive bioweapons. Because both of you are retired and no longer dependent on government grants, you can speak freely where most others cannot. So she sends an excerpt of an essay about this. It's the U.S. Army's research facility at Fort Detrick, which did indeed do offensive weapons research. 2001, Department of Homeland Security began promoting biological warfare research, and some stuff about dozens of new laboratories have sprung up to do biodefense studies, and let's see. Assertions that Fort Detrick's research agenda is clearly an offensive program, and goes on
basically
citing,
saying that we're doing bioweapons research,
right? Which would be a violation of the Biological Weapons Convention.
Do you know anything? I mean, it's hard to know about top secret stuff, but do you know
I could tell you, but
this?
then I'd have to kill you.
Yeah. Okay.
No.
You can try it over the Skype.
Yeah, it's like with anything like this, you can always hypothesize that there are top secret activities going on in the government that are nefarious in some way.
Yeah.
I am highly skeptical about claims that there is an actual offensive weapons program going on. It is not something that we have a whole lot of motivation to do right now,
Yeah.
and there's an enormous amount of downside if anybody found out about it.
I am truly skeptical myself. And the author of this article, the article is in truth-out.org, and the author is Patricia Hines. And I don't see anything in here about what her sources are.
Yeah,
I don't
that's
know
a
where
problem.
she's getting her information. But,
Yeah, there are a couple of little quotes that are from official documents, but they're not. They don't say anything about an actual offensive program. They don't really
Right.
prove anything.
Right.
So it's then just spinning these quotes into this idea that that there is such a thing, and also really what most of this stuff is about is that the biodefense research that's going on may not be currently conducted in the safest way possible, and there's some accusations that that these labs are not following necessarily correct procedures, that they haven't accounted very well for all of their stocks of biological agents, and that actually has some basis in fact. There have been incidents with some of these facilities that are a little bit disturbing, but they've come to light and been the subject of substantial disciplinary actions in most cases, I think. So yeah, I would certainly agree with the notion that we should all be a bit uneasy about vastly expanding the amount of research we do on potentially deadly pathogens, and make sure that these are very carefully controlled and that the labs that are working on them are operating at the highest standards. And it certainly behooves us to make sure that that's done as transparently as possible. But I'm not going to go with the whole conspiracy theory that this is an offensive weapons program and that everybody's up to no good.
Yeah. Well, we'll post this text and let readers have a look at it themselves. And I just would like to correct that I am not retired yet.
Right.
And still am dependent on government grants.
I'm not retired, but I'm not dependent on government grants.
Yeah.
Speaking of this, coincidentally, I just had recently an email from an ex-student here at UF who now works for Homeland Security with a news post from fredericknewspost.com saying that Department of Homeland Security, this is dated September 23rd, Department of Homeland Security lab at Fort Detrick passed its inspections Thursday and is ready to begin doing research with select agents. This is a BSL-4 facility.
Right.
And this article goes through all of the stuff, or some of the stuff. It highlights the stuff that they had to do to pass muster with the CDC in their inspections. And it's a sort of an interesting insight into the rigor of what they have to go through. So.
All right, let's do one more, and I think it's your turn, Rich.
Hi, TWiVers. I'd like to make a correction/comment to a statement made on TWiV 146 at about an hour. No one has ever purified, the comment is, quote, no one has ever purified MHC molecules and has done sequence analysis to see which sequences of HIV epitopes are in MHC molecules, end quote. Whilst it appears that no one has eluted peptides from HIV-infected cells, epitopes were eluted from MHC1 class molecules from cells infected with HIV recombinant vaccinia virus, see abstract below, and cells transfected with HIV DNA. And he attaches another paper. So it's not HIV-infected cells, but cells that through one mechanism or another are expressing HIV proteins. This field of research, combined with work on synthetic peptides, have confirmed the nature of the epitopes and the T cells that recognize them. The use of transfectants in vaccinia recombinants rather than HIV-infected CD4 T cells may have had practical reasons, since large quantities of infected cells are, or at least were, required to elute and identify the peptide epitopes. as anybody who spent a good part of his PhD growing large quantities of melanoma cells in order to elute and identify peptide epitopes recognized by cytotoxic T cells, I felt kind of strongly about this point. Anyway, suffice it to say, I really enjoy your podcast as I continuously learn a lot. Keep up the great work you're doing. Regards, Jorg. Jorg is in Germany, and he sent us a reference to these articles, which do indeed identify some MHC-bound peptides from HIV.
All right.
Cool.
So that's good to know.
Thank you. We are always happy to be corrected.
We're here to learn.
And
Here
we
to
only
learn.
we only have 2 left. We did pretty well.
Not bad.
Yeah.
That brings us to Science Picks of the Week. Let's start with Alan.
My pick, if you haven't seen it already, is a really cool stop-motion video. It's put together from a series of photographs that were taken automatically from the International Space Station. And NASA somebody at NASA went ahead and pasted these together into a video as the station is orbiting a part of the southern hemisphere. What's cool about it is that the station is orbiting across the dark side of the planet, and you can see the southern lights, Aurora Australis, from space as it goes over them, and it just looks really, really cool.
Nice.
It is amazing.
Yeah, it's gorgeous.
And actually, one of the things I noticed about this that I thought was interesting as well is that the solar panels on the space station are moving as this happens. They must automatically move to catch the best
Yes.
sun. Is that right?
Yeah. As the station moves through its orbit, the panels constantly adjust so that they're constantly looking at the sun.
Stuff is amazing, and it's going on all the time up to that is an amazing picture.
Cool. Very nice.
Good stuff.
Rich, what do you have?
What do I have? I have a link to a medical animation site, basically. This is one of their demonstration videos that was sent to me, I think, by Nassim. The company is called Ghost Productions, and they are illustrators of this stuff. And this is just one of their demonstration videos. You can go to their website and find more. But I thought that this was pretty entertaining. It's an animation of a guy who's painting the ceiling of the Sistine Chapel, and some janitor runs into his ladder, and so he falls down and breaks bones in about 10 different places in his body. And then they have this amazing animation that shows all of the different hardware that they can fix this guy up with all the screws and stuff associated with it, how they're put in. And it's first of all, the medicine is incredible, and the animation also is pretty incredible. And the other demos at the same site are pretty good. So
Very nice. That's very cool. Yep. My pick is called Science 360 Radio. It is a new website by the National Science Foundation. Basically, it has collected about 100 podcasts on science, and when you go to the website, one will always be playing. They rotate through their whole collection, or of course, you can go to the collection and pick what to listen to. And yours truly's, all 3 of our podcasts are listed, as well as Mark Pelletier's Futures in Biotech, Dr. Kiki's Science Hour. They're all there. And in fact, if you go there right now, while we're recording, it's playing This Week in Microbiology, episode 16.
Cool.
No kidding.
It's really neat, and they have an
So
app.
who's doing this?
NSF.
Okay.
In fact, Mark Pelletier told me about this a few months ago. He said it was in the planning, and it just launched. They have iPhone and Android apps, too, you can use to pull the content. This is really good. So they're going to get more people to listen to these podcasts. So
That's great.
we're here.
Right.
We're in it.
Yep.
So
Excellent.
check that out. And there are a lot of other science podcasts there as well.
We're on the dial.
We're on the dial. But, of course, listen first to TWiV, TWIP, and TWIM, and then move on to everything else.
Right.
And that will do it for TWiV 151. We, of course, are on iTunes at the Zune Marketplace, at microbeworld.org, or at twiv.tv. There's also an app produced by the American Society for Microbiology for your iPhone or Android device, and you can find that at microbeworld.org/app. We have now done most of the emails that are in the queue, so send us more.
Yeah.
TWiV@TWiV.tv. And don't forget our Facebook page at facebook.com/thisweekinvirology. Rich Condit is at the University of Florida, Gainesville. Thank you, Rich.
You're quite welcome. I feel privileged to be doing this. This is just a gas.
We are lucky to have you. Thanks,
Well,
as usual.
I just feel really lucky to be here. Alan, are we going to meet someday?
I certainly hope so. That would be great.
Someday we will have a
TWiV
live
party.
TWiV somewhere,
Yeah.
and all of us will be there.
And Dixon's going to cook, right?
I think so. I think we can go to some venue at some point that, and all of us are there. It'll work out. I'm working on it. But always great to have you. Alan Dove is at alandove.com. Good to have you back, Alan.
Always a pleasure.
And
I missed it.
We missed you, Alan.
Great.
Yes, we missed you as well, especially the 3rd anniversary last week. But you'll be here for the 4th anniversary.
I certainly hope so.
Hopefully. But you cannot predict the future.
No.
Can only hope. Good to have you back. I am Vincent Racaniello, and you can find me at virology.ws. You have been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.